Welcome back to the channel. Have you ever wondered how a bacteria cell which is only about 1 to 2 micrometer in size is able to accommodate DNA molecule which is 1 millimeter long? If this DNA remains stretched, it will be nearly 1000 times longer than the bacteria cell itself. So how the bacteria has solved this problem? So in today's video, we are going to understand how the DNA is packed in prokaryote with the help of diagram. By the end of this video, you will be also knowing why loop domains are important and why this topic is generally asked in the NEET. Now see, first of all, start with the bacteria. See, first of all, the bacteria DNA is double strand and it is circular, not linear. So you should know bacteria DNA is double strand and it is circular but not linear. They are present in a specialized region in the cytoplasm. That specialized region in the cytoplasm is known as nucleoid, which is not bounded by any membrane. Now, if you see the diagram very carefully, you are first finding it is going for the negative supercoiling. It is going for the negative supercoiling. Now, negative supercoiling means what? It is just, you can imagine for this, you can even imagine one imagination analogy. You think of twisting a telephone wire. When you twist, it coil upon itself and occupy less space. Same happen with the bacterial DNA. Negative supercoiling will shorten the DNA without changing its information. The enzyme which is responsible for introducing the negative supercoiling is DNA gyrase. This is the enzyme which is responsible for introducing the negative supercoiling. It generally belongs to topoisomerase family. It is very important because it continuously adds negative supercoiling, helping the DNA to remain compact and still allowing transcription and translation to occur. Now, see the next. It is making a supercoiling is going on. Twisting the DNA into negative supercoiling, what it will do, it will reduce the length, and because of which it can be packed into the proper loop. Now see here, this is the diagram of the loop domain. Now why these loop domains are important? And if you see carefully, the circular chromosome is folded into many loops. See here, so many loops are made. Nearly 50 to 100 loop domains are there, and each loop domain is attached to this center, this protein which I'll highlight with this color. See this color which I'm highlighting it. It is this one. Can you see now? This is the scaffold protein. This is a scaffold protein. So loop domain is the one which is attached to the protein, with that protein is known as scaffold protein. This is scaffold protein. Now why these loop domains are important? Just imagine you are trying to hold several rubber bands separately. If you twist one rubber band, others should remain unchanged. Similarly, each loop behaves as an independent domain. It means if one loop becomes supercoiled or relaxed, neighboring loops are not affected, which prevent excessive twisting from spreading throughout the chromosome. That is the major advantage of loop domain organization. So that this problem should not be, if one is going for relaxation, second should not go for that. Now, apart from this, some other proteins are also helping. Nucleoid-associated proteins, they are also helping. One is HU. HU, what it will do, it will bend the DNA and promote compactation. You can see here HU, it will bend the DNA and promote compactation. Second is HNS. It is the one which compacts the DNA and regulate gene expression. IHF, which bends DNA sharply, and FIS helps in DNA organization and transcription regulation. So these are few nucleoid associated proteins. They bend DNA, they stabilize the loop, and they maintain chromosome organization. You can just think these are the clips which keeps the folded DNA in the place. Without them, the chromosome will lose its compact organization. So please remember, in bacteria, DNA packing depend upon loop domain, it depend upon negative supercoiling, and it depend upon nucleoid-associated protein and not histone. Histones are not used. Why this packing is necessary? DNA should be packed so that the extreme long DNA can fit itself into the bacterial cell, which is small size. Second, independent loop domain will prevent the spreading of supercoiling. Then DNA can be assessed for easy replication, transcription, and repair. If DNA is too tightly packed, then this process cannot occur efficiently. So bacteria DNA packed to achieve the perfect balance, that is high compact but easy accessible. Please learn carefully. This is very important fact. These are the loop domains which are attached to the proteins. Clear? Many loop domains are there which are attached to the proteins. This is protein present. This is protein. Clear? So it is packed into the loop-like structure which are not that much tightly packed and easy accessibility for the replication or transcription. So once again, just revise it. Bacteria has one circular DNA which is packed with the help of the proteins to form a nucleoid structure. It is not inside the nucleus, it is in the nucleoid. It is making 50 to 100 loop domains, and each loop is attached to protein at loop anchor. You can see this is loop anchor. It is attached to the this one DNA at the loop anchor. DNA is compact with the help of negative supercoiling, which is introduced by DNA gyrase. Bacteria will not make nucleosome. It will not use histone proteins, but it will use nucleoid-associated histones, nucleoid-associated proteins. So if conclusion is the entire mechanism can be learned in one line. Circular chromosomes will make loop domain which is attached to loop anchor, negative supercoiling by DNA gyrase which is stabilized by nucleoid associated protein to form a compact structure inside nucleoid. If you find this explanation helpful, don't forget to like the video. Please share it with your friends and subscribe to the channel for more detailed biology concept explained in the easiest way. Thanks for watching and hope so you will be finding the topic very easy. And you can check down in the description the in every this course videos you will be finding MCQ theory plus MCQ is attached. So please check on it.